Good afternoon, everyone. I would like to welcome you to the fourth webinar of the Quality Webinar Series organized by EATRIS. My name is Rosanne Vechter and I'm Education Manager at EATRIS. I will be hosting this webinar today. Today, we are very pleased to have two speakers joining this webinar about pre-analytical validation and quality systems in biobanking. Before I introduce our speakers, I want to make you aware that this webinar is being recorded, including the questions and answers at the end of this session. The recording will be made available on EATRIS website shortly after this session. We have reserved some time at the end for questions. We invite you to write down your questions in the question box in the control panel, as you can see on this slide. First speaker for today is Dr. Monica Marchese. Dr. Monica Marchese holds a PhD in biology and pathology of aging from the University of Genoa, Italy. After receiving a fellowship from the federal government of Canada, she moved to Hamilton to collaborate and lead several projects at McMaster University. During her postdoc years, she has worked on animal models of neurodegeneration, including triple transgenic mouse models of Alzheimer's disease. Monica was then hired as laboratory manager for the Canadian Longitudinal Study of Aging, where she set up an automation-friendly laboratory equipped with a high-throughput platform. During her last 2 years in Canada, she focused on transferring and validating immunoassays from the bench to a robotic platform. In 2016, Monica has returned to Europe to be enrolled as biomarker validation scientist at the Integrated BioBank of Luxembourg, where she takes care of designing and executing biomarker validation studies for academia and Europe-based industry. Second speaker for today is Andrea Wutte. Since 2014, Andrea Wutte is leading BBMRI-ERIC Quality Management Service. The service includes quality management consultancy programs for biobank relevant standards and guidelines, monitoring and audit programs, training and education formats, assessment and the initiation of improvement strategy, respectively actions and accompanying measures. Andrea holds a European Quality Manager and Lead Auditor EOQ Personal Certificate. She acts as BBMRI-ERIC Liaison Officer to a number of ISO working groups. Andrea is a biologist by training. I will now hand over to our first speaker, Monica Marchese.
Good afternoon, everybody. Thank you very much for your nice introduction, Rosen. And as you said, my name is Monica Marchese. I work at IBBL, the Integrated BioBank of Luxembourg, and I take care of the biomarker validation service. I just want to give a brief overview of IBBL. IBBL is a not-for-profit public institute which is integrated with the Luxembourg Institute of Health. And our mission is to support the Luxembourgish biomedical research and to provide biospecimen-related services and biobanking infrastructure for applied medical research. IBBL is dedicated to quality. In fact, we operate under QMS as we are certified ISO 9001. This means that IBBL consistently provides services and products to meet regulatory and customer requirements. We are also accredited ISO 17025, and this accreditation pertains to technical competencies and is used generally by testing and calibrating laboratories. We're also ISO 17043 compliant, which means that we can deal with the development, organization, and distribution of proficiency testing schemes. I won't dwell too much on the description of this ISO because the next speaker in line, Dr. Andrea Wutte, is the specialist on this. I just want simply to mention that next year, in 2020, IBBL will seek the accreditation for ISO 20387, which is a really important document for biobanks, as it specifies the general requirements for competence, impartiality, and consistent operation, including QC requirements for the collection of biological material and associated data. IBBL offers a series of bioservices to a big group of partners in industry, academia, and European consortia. And as I mentioned before, I take care of the biomarker validation service. And if we look at the breakdown of our service, you can see actually the pre-analytics are a big staple in one of the steps of our service. So I would like to introduce the topic of pre-analytical validation and the importance of the pre-analytics by showing you a slide that I borrowed from the slide that normally I use to illustrate the concept of biomarker validation. And every year, as you know, the literature is enriched of new publication that report the discovery of new putative biomarker for a plethora of diseases. But unfortunately, only few of these biomarkers will make it to the patent office, and overall, a very small percentage of them will see the light of a clinical translation. So it's clear that many pitfalls and oversights can hamper biomarker translation. And the quality of the starting material, which is the sample, the biospecimen, is frequently one of them. In a seminal paper from 2007, Carraro and Plebani reported evidence that in a clinical laboratory, 3/4 of the sources of error occur during the pre-analytical phase. And this is a pie chart that describes the frequency of error in each phase of the so-called total testing process. So the pre-analytical phase, the analytical phase, and the post-analytical one. And a staggering 62% of errors actually occur during the pre-analytical phase. And interestingly enough, the literature referring to errors starts to appear already in 1947, but these errors that are reported in the literature are described only for the analytical phase. More recently, towards the end of the 80s, a new number of publications started to draw attention on the fact that error can happen also during the pre-analytical and the post-analytical one. So we should not forget that the samples for biomarker discovery frequently come from a clinical lab. So this 62% is really alarming. But in the majority of the cases, these samples come from the academic environment. So it's very easy to imagine what can happen to a sample coming from a research scenario where less strict quality or standardization procedure, if they're even present, are effectively put in place or enforced. So this is a very nice slide that I found from Lippi et al. Was published in 2011, which recapitulates the majority of in vivo pre-analytical errors that can happen in a clinical laboratory, and they are attributable to collection, handling, processing, and storage of biospecimen. And for each dot appearing on your screen, I would like to give you an example in the upcoming slides. So we should not also forget that there are not only pre-analytical errors that occur in the lab, but there's also the individual biological variability that contributes to the overall pre-analytical variation. We have contributors, which are so-called uncontrollable factors and controllable factors. Among the first one, the most common example is the age, or intra-individual variability factor. This factor is normally used to calculate the reference change value for each individual, and this is a value that is of utmost importance, especially when running projects related with the clinical validation. As we age, of course, the level of our analytes and the different esoteric biomarkers are shaped by the physiological and unfortunately pathological changes that are induced by the passing of time and by lifestyle habits that, of course, take the toll on each subject. Another example of uncontrollable factors is the intra-individual variability, and normally sex is the most common example. Levels of some analytes and biomarkers are known to vary between men and women. And in women, in the case of women, the inter-individual change overlaps with the intra-individual one during pregnancy and menopause. Then we have to consider the overall difference between individuals, which is really important when designing, for example, epidemiological studies. And this difference takes into account comprehensively age, sex, race, genetics, geographic habitat, and so forth. Here I pull an example that normally it's not frequently taken into account, that during studies, blood draws and bodily fluids are taken from patients that normally are lying down. And maybe because we are lying down due to a fractured leg, if we go and test some clinical biomarkers and some other esoterical biomarkers, we will see there are differences within the same subject when the blood or another bodily fluid is taken from this lying down position to the standing or the sitting one. And this happens because there's a shift of body water from the interstitial compartment to the vascular one. And of course, least but not last, stress is known to induce hormonal secretion of a large number of mediators, such as cortisol, prolactin, catecholamines, and so forth, that of course alter the pattern of the analytes in every subject. Among the controllable factors, we have the circadian rhythm, which are of utmost importance, for example, when testing pro-inflammatory mediators. So, for example, in the case of subjects with multiple sclerosis, one would have to be careful setting up a blood collection time to avoid finding significant but completely artifactual evidence when comparing MS subjects with the healthy subjects. And then, of course, we have fasting, diet, smoking habit, alcohol, and caffeine intake. For example, 200 milligram intake of caffeine will still induce 3 hours later after consumption of a cup of coffee an increase in renin and catecholamines. And fasting itself has been frequently associated with changes in many blood-based biomarker levels. Drug intake is another interesting controllable factor, especially when sampling individuals who are elderly. We know that sometimes elderly people can be really creative in the pattern of drug intake, so definitely this is a factor that needs to be taken into account. And finally, exercise is an example of controllable factor because it has been associated with changes in gene expression and changes in the methylation profile of different genes. And I will give you an example of this later on. So definitively attempts should be made to record information related to these variables so that later on when data are analyzed, one could control for these controllable and uncontrollable factors. So the STAR-B initiative is actually calling for standardization of timing and condition when collecting bodily fluids for subsequent biomarker analysis. And this is something that should be established long before starting a study. Doesn't matter what kind of study it is. So here I'm going to give you an example of how intra-individual variability can affect the performance of Mirna biomarkers. And in this case, the uncontrollable factor is age. Blood Mirnas are being progressively proposed as biomarker for different diseases. However, very little is known about the intra-individual variation of Mirnas, especially in healthy subjects. In this study, over a 1 year period, IBBL has measured the longitudinal variability of Mirnas by means of a wafer gene platform in blood derivatives and specific white blood cell subpopulation that were collected every 2-3 months. Now, we know from data published by other laboratories that the Mirna targets are pretty robust towards the most common in vitro preanalytical factors. But this has been shown to be valid only when the sample is taken once, only in a one-time period. IBBL results, as you can see here from this graph and this graph below, show that there is a continuum in the longitudinal variability of Mirnas in healthy subjects, presenting with cluster of very stable Mirnas, displaying a CV lower than 20–30%, and clusters of very unstable Mirnas with a CV higher than 130%. And additionally, IBBL data suggests that at least 20% of blood circulating Mirnas exhibit random individual variation with CV higher than 100% over time. So overall, these results suggest the importance of establishing platform-specific reference range intervals when considering subjects who are healthy versus subjects who are, for example, cases, and also the creation of a database for all Mirna's biological variability, as has been done already with the majority of clinical chemistry parameters. So this slide is an example of intra-individual variability where two pre-analytical factors overlap: age and exercise. One, as we said, is uncontrollable age, and the other one is controllable exercise. So the AAC gene has been found to be responsible for the secretion of two major pro-inflammatory mediators, IL-1 beta and IL-1 18. However, the same expression of this gene is regulated by methylation. And it's been observed that between young individual and elderly individual, there's a difference in the degree of methylation. So the more the AAC gene is methylated, the less chronic inflammation is present. So that explains why younger people normally are less inflamed in terms of chronic inflammation compared to elderly control. So in this study, a cohort of men and women underwent intense exercise training, and at the end, the degree of methylation of the ASC gene was measured. And what we can see actually is the degree of methylation increase in the elderly subject who were exercising compared to those who were not exercising, in a fashion that actually reverted the degree of methylation to a younger chronological age. So overall, these results suggest that one should take carefully into account the lifestyle as a preanalytical factor when, for example, designing studies encompassing age groups with individuals opting for physical activity versus subjects conducting a sedentary life. In this case, actually, we are considering a preanalytical factor that is more typical for the lab activity. DMSO is used as preservative agent when storing cells. And this figure is a snapshot from a publication showing the differentiation of stem cells, mouse stem cells, into embryoid bodies. And this model is frequently used to study the normal and abnormal development of mammalian cells. The differentiation of these cells is orchestrated epigenetically, as you can see here, by a set of loci. And the initial treatment of ESC cells with DMSO has been shown to alter a number of loci that become normally hypermethylated, hypomethylated, or they remain unchanged. And eventually, this change will translate in abnormal phenotypes in EB cells. So we have to keep in mind that it's really important to consider the kind of preservative used to store cells, especially when dealing with stem cells, to avoid aberrant methylation profiles that could lead to artifactual results. So this is a very elegant example. I really like this paper because it shows how known preanalytics of an analyte, so already published data on preanalytics of an analyte, can be exploited to validate a pre-analytical model for putative new protein biomarkers. So even before the discovery of new biomarkers, it would be important to identify those preanalytics that might compromise the power of the discovery itself. In this study, a panel of miRNAs and two proteins, TWIC and HGMB1, were proposed as biomarkers, novel biomarkers for cardiovascular disease. To assess their robustness to the most common preanalytical factors, researchers have established a preanalytical model that encompassed variables such as matrix type, collection tube, and anticoagulant, pre and post centrifugation delay, and so forth. So they took into consideration those parameters that normally we take into consideration when we perform a validation. In order to validate their models, they introduce concomitantly the measurement of interleukin-8, which is known to be sensitive to temperature associated with pre-centrifugation delay. Following the validation of their preanalytical models, the researcher concluded that matrix type undermined the robustness of the two biomarkers, while matrix type associated with transport temperature or centrifugation delays severely affected the miRNA panel robustness. So this is a very nice example of how studies should be planned before even being conducted. Having a biomarker for which, or an analyte for which the preanalyticals are known, and use it as a way to validate the preanalytical model of the two biomarkers that are part of the proof of concept. This is another paper that comes from IBBL and depicts a study that would aim at the investigation how tube preanalytics could affect RNA integrity. So we know the PAX-gene tubes and Tempus tubes contain stabilizers to avoid preanalytical mRNA degradation by endogenous endonucleases and to avoid the loss of specific mRNA due to unwanted downregulation and upregulation of events on certain genes. So at IBBL, we tested 16 different preanalytical conditions, including the ones here in bold character. They were part of the brochure provided by the manufacturer, and different assays were conducted to verify RNA quality and integrity. And you can see a really quick summary of the results that we found. Both collection tubes with their respective RNA extraction kit were deemed fit for the purpose for any downstream RNA analysis. However, the robustness of each blood collection tube to most common preanalytical variables, such as filling, effective inversion, temperature of temporary storage of the tube, alone or in combo, was different. So this is a very nice example of how one preanalytical or more preanalytical variables can affect the RNA integrity downstreams. And finally, as an example, I have this paper which shows that clinical analytes, and so in this case not esoteric biomarkers, can be used to investigate whether a sample has been compromised by preanalytical factors. So it's like a specific clinical analytes from the clinic are used to diagnose sample quality, and so they're like diagnostic tools that we can use to find out if a sample has been compromised or not. So in this case, researchers have exposed the sample to very extreme conditions, which of course are very uncommon for a clinical lab, but they become more frequent, for example, for samples that have been collected outside the clinical lab and they are stored in biobanks. So as you can see from the results summarized below, even small pre-centrifugation delay and minimal freeze-thaw cycle can compromise the stability, the concentration of many common clinical markers. So in order to compensate for all of these differences they found when comparing to normal laboratory condition, these researchers propose as corrective measures formulas to estimate, for example, the pre-centrifugation delay length, or to normalize concentrations when dealing with the more or less sensitive analytes. So are pre-analytics at the end of the day really important? Indeed, yes, because they have become part of the fabric of the new IVDR, of the new IVD regulation or IVDR. So on April the 5th, 2017, 2 new regulations were adopted. One is specific for medical device and will be enforced already as the new year, and the other one for in vitro diagnostic will be enforced, will become effective in 2022 after a period of grace of 5 years. It will replace the existing IVD Directive 9879 EC. So at the light of the new legislation and considering the definition given by personalized medicine, biomarkers, or not all of them, but a big part of them, will be deemed as a companion diagnostic, and as such will fall into class C within the new IVDR. This means that when a researcher, an SME, or an investigator will present the dossier to a notified body, they will have to provide the scientific viability for the biomarker, validity, sorry, of the biomarker. So they will have to present publication and or associated patent data. They will have to show the clinical performance of the biomarker, but most importantly, they will have to show the analytical performance of their biomarker, including the characterization of the pre-analytics for the samples that have been used for discovery and for validation, plus the traceability of values assigned to QC and or control materials. So things are becoming more and more strict for the CE marking of future products coming to the market, which of course encompass the use of a biomarker. And you can find more details here in this paper, and one of the authors of this paper is Dr. Andrea Wutte, who will speak right after me. So at the end of the day, the take-home message is not only in the biomarker field, but in general in the clinical, in the laboratory field, pre-analytics are important. They matter. One should check for the pre-analytics even before starting a study. Pre-analytics should be validated and should be considered all along in the lifetime of a biomarker. And since we started to talk about biomarker, I would like to conclude still talking about biomarker, just simply saying that there's a fil rouge that connects sample, biobanks, pre-analytics, and validation. And this fil rouge goes through the process of biomarker discovery and biomarker validation and application of the biomarker finally to the clinic via the process of translation. So in the case of biomarker discovery, of course, we want samples that are fit for the purpose, that has been discovered by means of only one SOP, possibly to strengthen the power of the discovery, that have been discovered using only one standard pre-analytical code. We want this sample to be accompanied by biological, clinical, and now we have learned pre-analytical data. And we would like to have this sample distributed by certified biobanks so that we know we achieve every single time consistency in our results. In the case of biomarker discovery, we have this kind of very strict scenario. In the case of biomarker validation, we can be less conservative. So we want still sample fit for the purpose, but we want them to be collected, processed, and stored via many SOPs and presented with different standard pre-analytical codes so that they can be validated by validated methods and by the use of standardized QCs. Of course, we want still data that encompass the biological, the clinical, and the pre-analytical characteristic of the samples. In this case, we would also add a requirement for data quality assurance. And in terms of origin, we would like for these samples to come from certified or accredited biobanks, so that consistency can be finally integrated with competency for the sake of good research, publication that talk about putative biomarker that will have a higher likelihood to become translated into the clinic. And with this, I conclude my presentation, and I look forward to your question at the end of the webinar. Thank you.
Thank you very much, Monica. I will now switch to our other speaker, Andrea.
First of all, I want to thank the organizer for inviting me to this webinar series. Thank you, Roseanne. And my task today is to talk about the quality system in biobanking. And Dr. Marchese already gave a very good introduction on how the validation and verification and quality standards in IBBL work. And I would like to give you a general overview about quality management system in biobanking. First, I would like to introduce BBMRI-ERIC to you. If for those who might not know BBMRI-ERIC, BBMRI-ERIC is also as EATRIS, a European research infrastructure. We are for the reason here to improve biobanking activities, and we bring together all the main players from the biobanking field, like researchers, biobankers, industry, and patients, to boost biomedical research. And we consist of, at the moment, 20+1 members and observer countries, and we are here to serve researchers and biobankers in the fields. In this case, I talk about quality management, but we also offer IT and LC services. Our main goal for the quality management is that we want researchers to be able to produce reliable findings. And with that, as a biobanker research infrastructure, we need to make sure that all researchers have access to samples and data of appropriate defined quality. And as a European research infrastructure, our ultimate goal is to make samples comparable across different countries and different biobanking and research systems. How do we do that? In this case, we set biobanking in the central role, but of course it is in the central role of BBMRI-ERIC and at the moment in this webinar, but it is part of a workflow in personalized medicine, in research and development. So we can see the biobanking field and in the quality management per se, there is a process input and a process output. And when we see biobanking in a workflow, then we can say that there is a starting point, and the starting point can be different starting points. Of course, the contact between doctor and the patient, it can be the contact point between researcher in academia with a research endeavor. But what happens then is that it set up a cascade of activities of different processes in the workflow, pre-analytical work processes, sample collection, sample preparation, processing. Then it comes to the laboratory, then it is in the laboratory environment processed and analyzed, and then we have pathologists who evaluate the different disease stages in a tissue, and we do have other sources like digital pathologies, other activities that are in this line. We have cohorts, we have different aspects, and all of these samples may end up in a biobank. And that puts biobanking in an important role because the storage and the processing in a biobank can start already at the doctor's office. It can continue over the different aspects that we show. The output of this entire workflow is on one hand, big data, on the other hand, publications. And what we need to know and what we need to oversee is that quality assurance and quality control in every single process step in this workflow has to be assured and controlled. And that makes it that the output of this process, as it is as Monica already told you, if this is a new device, a new in vitro diagnostic system that is created, or if it's a better treatment for a patient, that is our output. That is the environment where we are working in. So why do we use standards? Why is standardization important? And here I want to reach you as researchers. It can be a door opener to new fundings. When we see the last funding period, the Horizon 2020, we talk about money here. And you all want to have enough money for your projects, for your endeavors, for your new ideas to come to life, and so you apply for different projects. We have the IMI initiative, where we have 5.3 billion euro budget that is distributed to public-private partnership organizations, reaching out to academia and the industry. We, of course, all other stakeholders are involved in that process as well. And as you see, publication as one major output, and a lot of contribution is given to the patient. And we, in the light of the new Horizon Europe funding schemes, we talk about investing 100 billion euros into a funding pot that you would probably like to have benefit from. So as you know, there are 3 pillars in this funding scheme, and one of the pillars is health. And so we are definitely in this pillar, working in this pillar, and we are also in these excellent sciences involved. So, but what do we have to deliver? Of course, scientific output, scientific knowledge, but we also have to show that we have put all quality measures that are appropriate in place. When we see the need for quality management and research in research and development, we have to know as academia that we have to manage different expectation. In academia, scientific output publication is one of the most relevant output within a research career. But the academia also have to manage different expectations from interested parties. Interested parties can be, of course, the patient. It can be the manufacturer. It can be the industry. So keeping this in mind, it's we cannot see only academia as one site. We also have to see academia in different countries, in different organizations. So we have different activities going on. And we face different interested parties in different countries and different organizations. So what we have to deal with as an academia partner is quite heavy, I would say. So why, and here I would like to go back to this interesting idea. Why not taking advantage of standards that are currently available for biomolecular and biomedical research, but also for biobanking and other activities that we are working on. In this slide, I would like to give you a fruit for your thought. Standardization, standards is not the main issue and the main topic that researchers in academia are talking about or are familiar with. But I think the academia should get a little bit more closer to the idea that standards can help them to improve their own activities. So therefore, I tried to make here a cloud with terms where we see two aspects: the scientific publication in comparison to an international standard, and is this at all comparable? So what I wanted to show with this slide is that in the academia world, the scientific communication is a scientific publication. It goes into, or it shows new medical approaches. It deals with new hypotheses, puts into documented information, experiment studies and trials. It tries to minimize variability, and here already Monica has shown you the activities and the biological variabilities that a researcher deals with every day. Verification and validation is important for academia. Establishing new methods, going into new experiments to boost biomedical research, to go into biomarker research and output a peer review publication. On the other hand, an international standard, which is the main focus of a standard, is standardization, of course. But you would, as academia, you would always put, or you might put a standard into the light of business, new markets, cost saving. In international standards, quality, improvement, plan, do, check, act cycles are one important aspect. Interoperability, efficiency, due diligence. In standards, we talk about processes. We want to be able to transfer processes from one organization to the other organization in a very easy way. We talk about technical specification. We talk about fit for purpose, intended use, benchmark. We talk about customers, customer satisfaction. We talk about certification, accreditation, and audits. So we have maybe two words, different words, where we would like to see those melting into each other. So what I want to emphasize is to academia to take advantage of standards. And how can you do that? So international standards are not SOPs. International standards are a guideline, is a standard that gives you a minimum set of requirements for a specific intended use. For biobanking, the new biobanking standard ISO 23387, the requirements for biobanking, for the competence, of course, for the impartiality, but also for the consistent operation of biobanks, and also for quality control requirements to ensure that the biological material and associated data collections are of appropriate quality. This is handled in this document. Quality Management System Requirements, called ISO 9001-2015, gives a general view on how you manage your system in a way that the output is customer friendly, gives customer satisfaction, and you can deliver what you promise. Then there are other international standards relevant for the biomedical research field. Molecular in vitro diagnostic examinations, specifications for pre-examination processes for different specimens with a specific intended use to isolate RNA, DNA, proteins. And then there is another standard guidelines for auditing management system, which gives you an idea how you audit your management system to show compliance to your system. Oh, sorry, that came back to that slide. There are other international standards for laboratory practices. One is the known 5189/2012. It's for the medical laboratories. It shows the requirements for quality and competence in medical laboratories. And we do have the standard, the general requirements for the competence of testing and calibration laboratories. And there is another standard that can help you to comply to requirements for collection, transport, receipt, and handling of samples, called ISO Technical Specification 20658. Maybe you wonder about these short names. The short names are very typical in the quality management world because you don't want to rephrase the long name of the standard all the time. So usually you work in the quality management world only with the short acronyms. I know this is a little bit boring, but we also have standards on data quality. You can see on this slide that there are requirements expressed in how to work with master data, the exchange of characteristic data in syntax, semantic, encoding, and conformance to data specification, to provenance data, to accuracy, to completeness. So there are standards in place that you can get hold of, as well as the data quality management framework when you deal with data. How you can improve your information and data quality on concepts and measuring process reference models, and also the information about data quality management process assessments. I will go a little bit more into the detail how a standard is built up with the quality management standard for biobanking. We also have, and this is probably also important for you, security techniques, information technology, information security management systems, as well as code of practice for information security controls based on a specific standard for cloud services. So these are standards that you can get hold of in different organizations, and these standards are usually 10 to 15 pages, and you can look at these standards in a way that you can compare what is your current process, how is your current process look like in comparison to the requirements expressed in these standards, so you can easily, or maybe not so easily, but you can improve your own management systems. For the biobanking standard, we have the 2387. A standard for biobanking is in general built up of any standard is built up like this biobanking standard. It starts with the definition of a scope. In which scope does this are these requirements expressed for? Some connecting dots to other standards that are available that might refer that are normative. You should and you shall implement them, and you shall be aware of other standards here as well, but they point you at that direction. Very important, it is expressed the terms and definition so that we talk the same language when we talk about samples, when we talk about biobank, what is it? And then it comes to the more technical aspect, so the key processes of a biobank, where it is expressed in the general requirements, the structural requirements, the resource requirements, process requirements, and then also how shall be your quality management built up in a biobanking ecosystem. It would be too long to go into details of these different chapters, but what I want to point out today, because it fits to the presentation of Dr. Marchese, is the process requirements. In the process requirements, we have, okay, this is the web conference here. In the process requirements, we point on this direction. This is the publication that already Dr. Marchese was pointing at, that pre-analytical errors still account for nearly 60 to 70% of all problems occurring in the lab, and most of them are due to mishandling procedures during collection, handling, and preparing and storing the specimens. So what we are doing since then is to improve and to raise the awareness of this problem so that academia can improve their own processes. The biobanking standard is one step towards improvement, and we also have different other aspects that point in same directions. So the pre-analytical error as a fact, the irreproducible research, and economic loss that can happen because of that. So what happens in the past is that a EU project called Speedia was initiated to develop different standards. In this case, this was a European standard that point exactly in this direction to improve pre-examination processes. So that means the processes are described, and requirements for these processes are described in international standards. And here we at the moment do have 9 standards available for different specimen types with different intended uses. And we have standards in place for frozen tissue, to how to prepare and process frozen tissue so that we can isolate RNA or proteins out of these tissue sets. Formalin fixed paraffin embedded tissues for the intended use to isolate RNA, proteins, and DNA. How to process venous whole blood is outlined in 3 standards for the intended use to isolate cellular RNA, isolate genomic DNA, and isolate circulating cell-free DNA from plasma. We do have a European standard, it's called CEN standard, for metabolomics in urine, serum, and plasma. And as a good standard, the medical laboratories requirements for collection, transport, receipt, and handling of samples. At the bottom, you can see how these standards look like. We also refer on our website to these pages. We have set up self-assessment surveys to look if the organization can assess the quality of specific sample collections according to these international standards. The content of these standards is in all standards has the same pattern. It starts with the scope, also the normative reference terms and definition. Then it starts in the chapter outside the laboratory, how the sample specimen is collected and shall be collected, the sample donor sampling process and sampling and transport is described, and requirements are presented in that standard. Then how it is processed inside the laboratory, how the sample should be received, fixated, the evaluation of the pathology is described in this standard. If there is the need for post-fixation, embedding, aliquoting, storage, isolation processes is also described in this standard. It also points out when you're using commercial kits or if you're using laboratory-owned protocols, and the assessment, the quantity and quality assessment and storage of isolated RNA, DNA, and other molecules. Then the quality control of RNA, DNA, and proteins is expressed in the standards, and the impact of pre-analytical workflow steps on specimen quality, time dependency, of analyte integrity. We have, based on this very successful first European project called SPEDIA, currently ongoing, and IBBL is an important partner in this project, together with 18 other partners coming from different countries, from different organizations and academia. This consortium is working on new standards. They are in the pipeline, one can say, will be finalized in the next year, and then we can expect new standards for venous whole blood circulating tumor and organ cells, venous whole blood exosomes, saliva, frozen tissues, urine and other body fluids, fine needle aspirates, saliva and stool microbiomes, and FFPE tissue in situ staining procedure. So then you can see we have a set of international standards in place that can help you to improve the quality of your research and scientific work. Of course, it's an add-on to your scientific approaches, but I would really emphasize to take this standard into your consideration, to review it, and find the way to add your academic excellence. Yes, adding standardization and standards to your academic excellence. To keep you all updated on new and upcoming standards, we, as BBMRI-ERIC, we have a liaison to the ISO Technical Committee, where international standards are developed. The ISO Technical Committee 276 for biotechnology is still ongoing. Different standards will come up in the next few years. These clinical laboratory testing and in vitro diagnostic test system technical committee is dealing with this international standards for pre-analytical processes, which are per se important for in vitro diagnostic regulation, helps the manufacturer to assess the quality of the samples. So you see there is a cycle that can help you to also to deal with the needs of interested parties like industry partners, manufacturers, or if you yourself are developing new assays, then you should get hold of it. Where can you find tools and the information of which standard is relevant to biomedical research, to biobanking? Please go into the BBMRI-ERIC website. Here we have under service and support the subchapter quality management. Here you can find all needed information and links to the standard. Of course, you can contact us, and we are here to support you and to give you insight into these standards more in detail than it was possible today in this presentation. When you want to contact us, please, you know where we are, and please just feel free to contact us. And for the next year, we will again plan training seminars on these pre-analytical standards, and you all are very invited to this program. So for this, I think this was my last slide. Thank you very much, and I'm here for questions.
Thank you very much, Andrea and Monica, for your excellent presentations. We have now time for questions. So the first question is for Monica. How can we document pre-analytical factors?
So there's actually a tool which is called SPREC. I mentioned in my last slide, in the standard analytical code. And normally this is a 7-element code that corresponds to those that are considered the most clinical analytical variables. And this SPREC is constantly updated. It started in 2009, and now we are already at version 3. And normally it helps to identify all of those analytical variables that accompany a biospecimen. And this analytical variable can be already known, or can be maybe an indication for the biobankers to apply SOP and certain procedures so that you know, the sample can be treated according to the pre-analytics that it presents with.
Okay, and why does biomarker validation require more than one SOP, as you mentioned in slide 16?
Yes, so during the discovery, I mentioned that we want only one SOP, because if a project is conducted in terms of discovery from beginning to end following an SOP, that can only increase the power of discovery, because the number of variables that can affect the discovery are reduced to the minimum level. When we validate, of course, we need to put the biomarker robustness to the test, aside, of course, testing the fitness of the purpose of the method that is being used to validate it. So in a way, this sounds a little bit rough to say, but we want to test all the variables that can break the biomarker, and therefore we need to consider as many as pre-analytical variables that can present, which of course also that are meaningful. We wouldn't go and look for tubes or storage condition that would not make any sense. But of course, when we validate, we test the biomarkers, trying to encompass all the pre-analytical variables that are meaningful, so that in this case, on our side, we tend to not only give power to the proof of concept, but also to consolidate it according to what are the diktat of the biomarker validation process.
Great, thank you, Monica. I also have some questions for Andrea. So where can people find the standards, and what are the costs associated to those?
So the standards can be found at the website of the International Organization for Standardization, ISO.org. But you can also go into the BBMRI-ERIC website. Here we have it brought together all these relevant standards for biomedical research. And the cost of such a standard is approximately around 100 euros per document, and it includes approximately 20 pages, and you get a PDF for it.
Okay, great. And where can people find or get training on the standards that you've presented?
So as as you do as a IATRIS, you are doing training and education. So BBMRI-ERIC is also doing training and education, and we have on our agenda for next year more detailed training and education on these specific standards. Of course, you can also ask for a training, a customized training for a specific standard. This is also what we can offer to organizations. And for this biobanking training and education course that we are currently running, we have 3 courses running next year in January, and we plan to follow up on this biobanking standard in a new training as well.
Okay, great. And one final question, is the standard for biobanking for certification or for accreditation?
So the biobanking standard is for accreditation, because the differences is that in an accredited environment you have to show competence, impartiality, and also quality control and quality assurance measures. And therefore this biobanking standard is a standard for accreditation. So 9001, the quality management system standard, is for certification, where you don't have to show directly impartiality, but also of course competence in a way, but not in that deep way as it is with the biobanking standard.
Okay, great. Thank you so much. We have now come to the end of this webinar. I would like to thank you all for attending today's session, with a special thanks to our lecturers for their excellent presentations. Should you still have any further questions to one of the speakers, we welcome you to submit to us, and we will get back to you as soon as possible. The recording of this webinar will be made available to anyone interested on our website within 2 weeks. And I would also like to point out that we will have another webinar in 1 month's time regarding technology platform reproducibility. We hope to all see you again then. Thank you and have a lovely day.